Welcome to HD101, a free CME course for primary care providers, family practitioners, neurologists, and psychiatrists not familiar with Huntington's disease. This 10-part course is brought to you by the Huntington's Disease Society of America and We Move. Each session offers one ACCME credit per completed module for a total of 10 credits. To earn a credit, you must answer 75% of the post-content questions correctly and complete the evaluation at the end of each module. Please follow the links shown at the end of each session. We'd like to take a moment to thank our sponsors, the Griffin Foundation and Lundbeck, for their unrestricted educational grants which made this course possible. Before I introduce our speaker, I'd like to go over how to ask a question. Questions will be answered after the presentation. However, you may send a question at any time during the presentation. To send a question, go to the control panel that appears on the right side of your screen. There will be a panel for typing a question. After typing your question or questions, hit the send button. Dr. Bird will answer each in the order received. Module 2 will be presented by Dr. Thomas Bird, Professor of Neurology and Medical Genetics at the University of Washington. Dr. Bird has a long research and clinical interest in hereditary diseases of the nervous system. He is director of the HDSA Center of Excellence at the University of Washington. And now I'll turn the presentation over to Dr. Bird.
I'm Dr. Tom Bird from the University of Washington in Seattle. I'm director of the HDSA Center of Excellence at the University of Washington. I'm here to talk about genetic factors in Huntington's disease. These are my disclosures. And I'd like to take a few minutes and review genetic factors in Huntington's disease, starting with the history, and then move forward to the amazing molecular findings that have happened in the last couple of decades. It's fascinating to realize that more than 100 years ago, the autosomal dominant inheritance pattern of Huntington's disease was very nicely summarized by Dr. George Huntington, in fact, in 1872. He proposed a genetic mode of inheritance for this disease that really confirmed in a human disease what Gregor Mendel, the founder of genetics, was describing in pea plants at about the same time. Here are pictures of three contemporaries in the mid-19th century, Charles Darwin, Gregor Mendel, and George Huntington. They were all working at the same time, relatively oblivious to what each other were finding. But Huntington actually described dominant inheritance in a human disease, and Gregor Mendel had described it in pea plants for the first time. What do we mean by autosomal dominant inheritance? The characteristics are that multiple generations are affected. That if you have Huntington's disease, the risk to each of your children is 50%. Males and females are equally affected, and it is called fully penetrant. That means if you have the mutation for Huntington's disease, you will develop the disease if you live a full lifespan. If you do not have the mutation, you will not develop the disease, and you cannot transmit it to your children or grandchildren. So let's take a few minutes and discuss that word penetrance I used a few moments ago. What do we mean by penetrance in a genetic disease? It refers to the proportion of carriers of the mutation who express signs of the disease. Fully penetrant means 100% of people carrying a mutation will eventually show signs of the disease. In Huntington's disease, it's age dependent. That means there's often a delayed onset of expression of the disease, so that at age 20, about 5% of people who carry the mutation will show the disease, but by age 40, half the people, 50%, will already have shown signs of the disease, and by age 65, at least 95% of people carrying the mutation will show the disease. This is demonstrated in the graph on this next slide. You can see that the average age of onset of Huntington's disease is about 40. And there's a spread of onset both earlier and later than age 40. But if you carry the mutation by age 65, the vast majority of people will have shown signs of it. And if you live a full lifespan to age 80, everyone who carries the Huntington mutation will show signs of the disease. This is the kind of risk that people have to undergo when they are born to a parent who has Huntington's disease. The kind of risk that everyone had to deal with before genetic testing was available. The graph shows that when you're born, your risk is 50% of having the disease, and you would have to wait until you were in your 50s, 60s, and 70s before you could be certain that you had not inherited the gene. This reflects the state of the art before the advent of genetic testing. I've also used the term expression. That's different from penetrance. Expression refers to the signs and symptoms of a disease. They're so-called test-dependent. That means if you're talking about symptoms, it's expression of symptoms. If you're talking about signs on a physical exam, it's expression of signs. If you're talking about expression for neuroimaging, it would be whether or not you see signs of brain atrophy or caudate atrophy on a CAT scan or an MRI scan. For many diseases, there are biochemical tests that are available that could also be included in expression of the disease. There are no biochemical tests for Huntington's disease at the present time. And the expression of Huntington's disease is variable. You can have motor symptoms, that's the chorea and the involuntary movements. You can have behavioral problems with personality. You can have cognitive problems with thinking and attention and executive functions. These are the various expressions of Huntington's disease, and some people will mostly have one, some people may have two, or some people may have all three. That's the variable expression. The major genetic advance after Huntington's description of the disease actually occurred more than 100 years later. In 1983, the region of the human chromosome carrying the gene for Huntington's disease was discovered. It turned out to be what's called the short arm of number 4 chromosome. And this was able to be discovered because of a huge population of persons with Huntington's disease living in Venezuela, so-called founder effect, where there was a large village containing many persons all related to each other who had inherited the gene for Huntington. This is a figure that shows the human chromosome number 4, and you will see way at the top of the short arm is the location of the Huntington gene. It actually was difficult to locate because it was way out at the tip of this chromosome, making it hard to find genetic markers in the vicinity. But it was eventually discovered in 1983. It took a decade, 10 more years, for the actual gene to be identified, and that was in 1993. And quite surprising, it turned out that it was a disease where the gene contained a DNA repeat expansion, referred to as a CAG repeat expansion. CAG refers to the 3 DNA chemicals in this region, cytosine, adenine, and guanine, that code for an amino acid called glutamine. And it was discovered that it was a repeat of this CAG over and over again above that occurs normally in the population that seemed to be causing Huntington's disease. And we'll talk a bit more about these various repeat expansions in a moment. It also became recognized within a few years that Huntington was just one of many diseases, mostly of the brain, that are caused by these CAG repeat expansions. This slide shows several of those diseases. Many of them are called hereditary cerebellar ataxias. So Huntington's disease joined this group of degenerative genetic diseases of the brain that were caused by these repeat DNA expansions in the relevant gene. Exactly how that expansion occurs still is not entirely understood, but this slide is meant to show that when DNA is replicated or when it's repaired, the ability to do that is not always perfect, and sometimes there's a little fold that occurs, and that extra piece of DNA gets incorporated into the gene that is being replicated, and that extra piece becomes the CAG repeat expansion. This graph, published by Dr. Nance, shows the distribution of these CAG repeat expansions in the normal population and in the group of patients with Huntington disease. You'll see on the left that we all carry a CAG repeat in our normal Huntington gene, and that typically varies from 10 to 20 CAGs. That's perfectly normal. It causes no disease at all. In the Huntington population, this expansion is enlarged, so it becomes greater than 35 or 40, and this is associated with Huntington disease. You can see that it trails out into fairly large expansions, and we'll describe in a moment how that can be associated with juvenile Huntington disease. So back to the issue of penetrance, it now becomes a little bit more complicated. People with Huntington's disease have a CAG repeat expansion that's 40 CAGs or greater. If you fall into that group, you will develop Huntington disease if you live a normal lifespan. In fact, if that CAG repeat expansion is 60 or more, you're likely to develop the disease fairly early in life, sometimes even before the age of 20, which is juvenile Huntington disease. However, there are a couple other categories to pay attention to. One is the CAG repeat expansion between 35 and 39. This is abnormal, and it's referred to as the range of decreased penetrance. Some persons with an expansion in this range develop Huntington's disease, and some do not, therefore decreased penetrance. However, their children are at 50% risk of inheriting the Huntington mutation. There's even another category, 28 to 34 CAG repeats. This is referred to as the unstable range. Persons in this range do not develop Huntington's disease, but that mutation area is unstable, and their children are at risk for developing a larger mutation and the disease. This next slide illustrates this relationship in more detail. The x-axis at the bottom shows the size of the CAG repeat, and the y vertical axis shows the age of onset. First, you should notice that there clearly is a correlation between the size of the CAG repeat and the age of onset. So the larger repeats are associated with earlier age of onset. You'll see on this slide that 76 or 77 is the largest expansion in this group of 360 patients. In fact, a few expansions even larger than that have been described. We've seen one individual at the University of Washington who was a child who had 165 repeat expansions and had onset of the disease at age 3. So clearly there's a correlation between the size of the repeat and the age at onset. But the other thing to notice is that particularly in the range in which most patients fall, the correlation is not very good. For example, if you had a CAG repeat of 45, you can see that some persons had onset in their 20s, other persons didn't have onset until they were in their 50s or 60s. So in general, the repeat expansion number cannot be used in an individual person to predict the age of onset. What about the phrase anticipation? This refers for a trend for earlier onset with each generation of a disease. And in the CAG repeat expansion diseases, it's correlated with the size of the expansion, as we just demonstrated, such that very large expansions are associated with juvenile onset. In Huntington's disease, juvenile onset is more common when the father is the affected individual. That's so-called paternal transmission of the disease. The reason for that is not entirely understood, but it appears that CAG expansions are more unstable during spermatogenesis, the development of sperm. However, I point out that not every case of juvenile Huntington's has a father as the affected parent. We have seen several families where it has been the mother. So the size of the expansion in the parent has a great deal to do with the instability of the mutation. Let's take a minute and discuss some of the reasons you might want to do a Huntington genetic test and some of the reasons you might not. First of all, there are medical reasons for testing, and these are the reasons that are usually on the mind of the physicians involved with the family. For one thing, the genetic test for Huntington is highly specific. It's considered the gold standard for the diagnosis of the disease. It's basically part of the definition of the disease. If a physician has an individual with chorea and is trying to determine if that's Huntington's disease or not, the genetic test will give the answer. If it's positive, if there's a big CAG repeat expansion, then you've made the diagnosis of Huntington's disease. If the test is negative, that allows the physician to search for other diseases. One can exclude the diagnosis of Huntington's and move on to the differential diagnosis of other conditions. The test also gives some idea about prognosis. As we've talked about, the disease can be more severe with very large repeat expansions. And of course, the test is very important for genetic counseling. It's how we decide who is at risk for the disease in the family and what that risk is. Another theoretical reason for doing the testing in the future is for the potential for early intervention. If there are treatments or cures or preventatives for this disease, the idea obviously would be to intervene as soon as possible, and so you would want to identify people with the mutation as soon as possible. At the moment, we don't have a prevention for Huntington, but this is something that may become more important in the future. It's interesting to step back and recognize that the reasons families and patients decide to have the genetic test is not always the same reasons in the mind of the physician. The family members often come to genetic counselors and say they simply want to know, do I have this disease gene or not? They're individuals who can go through life and not care, not worry, not want to know, and decide not to do the testing. In fact, that's the majority of family members with Huntington's disease. But there are persons in the family who come to physicians and counselors and say, I need to know. They seem to be driven by stress and anxiety of not knowing, the ambiguity of not knowing, and they say that they would be less stressed knowing the answer rather than living each day not knowing. Obviously, persons also sometimes would like to know for family planning reasons, for career decisions, for travel and financial plans. Those are all reasons that are given for deciding to do the genetic testing. And of course, people hope they have not inherited the mutation, will not develop the disease, and can get on with the rest of their life without the worry of having the HD gene. One has to pause, however, before doing the test. A positive test could increase someone's depression rather than decreasing it. It could increase someone's feelings of anxiety rather than decreasing it. We worry that persons who are positive for the test may just give up and say, well, my life is now set. I'm going to get this disease. Why should I bother doing anything more? In fact, some persons may consider ending their life and committing suicide. That's a huge worry in Huntington's. Fortunately, it's not frequent, but it's something to consider. It's also important to note that persons who have a normal test result, a negative test result, do not always feel that they're off the hook. There's a condition called survivor guilt, where they wonder why were they the person in the family that didn't get the disease, and who's going to take care of their other affected family members. They feel that burden almost as much as if they had had a positive test. There's a genetic fallout in families when you have a positive test, and by that I refer to the effect on other family members. Suddenly the issue becomes who else is at risk, who's affected with this disease and who's not. Who should you tell the test result to? Do you tell your other family members or not? How far will this information spread through the family? It's sort of a ripple effect if you look at the family. Someone gets the diagnosis of Huntington's with a genetic test, and slowly brothers and sisters and children and parents and aunts and uncles and cousins learn about this positive test result. Sometimes it's not a ripple, sometimes it's a bombshell, and it's as if a genetic explosion has gone off in the family, and there's a lot of consternation and anxiety that immediately becomes apparent. Another issue is the diagnosis of Huntington disease in children, particularly should children at risk be tested. Who decides that? Does the child decide? Does the parents decide? Does the physician decide? What would be the long-range effects on a child of knowing that they had inherited the mutation for Huntington, even though they were perfectly normal during childhood? There's an entity called the self-fulfilling prophecy. If people around a child know that child has the mutation for Huntington, will they treat the child differently? When the child has problems, will the family and friends and teachers assume that's due to the Huntington gene rather than the normal effects of growing up as a child? That's a serious issue that we try to avoid. And for that reason, the general policy is not to test children until they reach the age of 18, which is the legal age of adulthood in this country, and let those children who attain adulthood decide for themselves. The genetic testing for Huntington also sometimes arises during adoption. Should a child put up for adoption who's at risk for Huntington be tested? The answer is usually not. There's no good reason to do that. It's the same as testing any child. You should wait until they're adults. Should the adoption agencies tell adopting parents that the child is at risk for Huntington disease? I feel they should. It's something that should be known about a child's family history. What about social discrimination when someone has a positive genetic test? Are we creating a group of genetic social outcasts with this genetic testing? Could a person lose their job because they have a positive genetic test? Could they have difficulty finding a job? Will they have trouble getting insurance? It's good to know that a federal law was passed in 2009 called GINA, the Genetic Information and Non-Discrimination Act. And this basically says that you cannot withhold health insurance from a person who has had a positive genetic test. That means all individuals who have tested positive for Huntington's are still eligible for health insurance, and their premiums cannot be raised on the basis of that genetic test. However, it's important to note that this does not stand for life insurance or disability insurance or long-term care insurance. A person who has tested positive for the Huntington mutation is very likely to have difficulty getting those kinds of insurance. Think about the effects of this genetic testing on employment. What if an airline pilot or a physician or a company executive or school teachers or someone in prison has a positive genetic test for Huntington's disease? How will that affect their employment? What will their employers think if they find out? These are not simple questions and should be taken into consideration before people rush into genetic testing. It's also important to ask who wants the genetic test done? Is there a legal reason? Is there a prosecuting attorney or a defense attorney that's asking for this result? Is the individual in jail or in prison and the prison's asking? Is there a divorce proceeding that will be impacted by this result, particularly one involving custody of a child? Is the individual on active duty with the military? Is it an insurance company that's asking? Is it an employer that's asking? Find out why these tests are being requested before jumping to do them. So now let's take a brief look at the Huntington gene, since we're talking about the genetics of this disease. What is this gene? In genetic terms, it's a fairly large gene. It has 67 exons. Those are the parts of the gene that code for a protein. That's 200 kilobases long, and that Cag repeat expansion is at the very beginning of the gene in exon 1. And the Cag codes for an amino acid called glutamine. So the result of the expansion is to have a very long line of glutamine amino acids in the protein made by this gene. There are actually two forms of the gene that are expressed by the nuclear DNA. Here's a cartoon showing the Huntington protein and showing you that it's more than 3,000 amino acid long. And that poly Q at the very beginning of the protein, Q stands for glutamine, and this is the multiple stretches of glutamine that are coded for by the Cag expansion. The protein made by the Huntington gene is primarily in the cytoplasm of the cell, and it occurs in all cells in many organs of the body, so it's said to be widely expressed. We noted that it had more than 3,000 amino acids. The normal function of this protein is not entirely clear. It's probably involved in transporting and trafficking other proteins and chemicals in the cytoplasm of the cell. The abnormal function is also not entirely clear. It's said to be a cytotoxic gain of function, meaning that the protein is active and doing something, but that function is toxic or damaging to the neurons. It's also noted that the protein shows up in the nucleus. Remember, I said it's usually in the cytoplasm, but it gets into the nucleus partly because it's cleaved or cut into smaller pieces in the cytoplasm and leaks into the nucleus, where it may do additional damage. The glutamines that are coded by the gene can sometimes cause toxicity to neurons. So there's this theory of glutamate excitotoxicity that may be playing a role. Mitochondria, which are the energy packets in all cells, are probably abnormally functioning in this disease, adding another component to the toxicity. And the repeat expansion varies a bit from organ to organ, but its major component of toxicity is obviously in the brain. This is a microscopic slide showing a human nerve cell, and the center of this cell is the nucleus, and the black arrow is pointing to the aggregate of the Huntington protein, the inclusion called aggregating in the center of the nucleus of this cell. This can be seen under the microscope with certain stains from the brains of individuals who have died with Huntington. And whether this is causing toxicity or not is not clear, but it's an abnormal finding under the microscope. It's important to know that there are genetic models of Huntington disease. The most common and most useful one is in mice. If you completely eliminate the Huntington gene from a mouse, it's lethal. The mice do not survive the embryonic stage. That clearly shows that this protein produced by the Huntington gene must be extremely important and contributes to the life of the animal. Without it, the animal dies. If, however, you have a normal mouse that you insert the abnormal human gene into the mouse's gene, the mouse survives, but it develops disease. One of these is called the R62 mouse. It has the human exon 1 with the Cag repeat expansion in the mouse gene. These mice gradually develop weakness, poor balance, weight loss, and have a shortened lifespan. Microscopically, they have those intranuclear inclusions in the neurons of the nerve cells. So this looks like a good mouse model for Huntington's disease. Some of them develop diabetes, and there is an interesting range of research suggesting that there may be a connection between developing diabetes in individuals with Huntington's disease, and that happens in some of the mice. So this mouse is considered the most important genetic animal model of Huntington's disease, and the experiments trying to find treatments and preventatives for Huntington's are largely done on these mice. So finally, it should be realized that this knowledge of genetics in Huntington's, although it's extensive and really advanced in the last 2 decades, has still left a lot of unanswered questions. What's the normal function of the Huntington protein? What's the abnormal function that's caused by the CAG expansion? And why is it that that CAG expansion causes toxicity? And what's the role of those intranuclear inclusions? Are they toxic? Are they protective? Are they necessary in the disease? These are questions that will be useful to have answered in the future. And why is it that there are certain neurons in the brain that are especially vulnerable to the toxicity of Huntington's disease? There are neurons called medium spiny neurons in the caudate nucleus of the human brain, and they are especially sensitive to the toxicity of this mutant protein. Why is that? And will that help us learn how to prevent, stop, and reverse the molecular pathology of this disease, which would result in preventions and treatments? And that's why understanding the molecular events in this disease will be crucial in leading down the path of prevention and cure of Huntington's disease.
In just a few minutes, we'll begin our question and answer session. Please take a moment to review the resource slide for this module, followed by instructions on how to complete the post-content test and evaluation to obtain your CME credit for module number 2. An email with these instructions will also be sent to you tomorrow. So let's start with a few questions. One is, do you ever do genetic testing on children? During the presentation, I made a point that you usually do not test children, and that could be detrimental to the growth and development of the child, particularly socially and emotionally. There are exceptions, and the major exception is if you suspect juvenile Huntington's disease. So if the child is beginning to have changes suggesting the juvenile onset of HD, then it makes sense to test the child to see if in fact they have the repeat expansion that correlates with the disease, because the family, the school, the persons caring for this child need to be aware of whether the child has juvenile Huntington's or not, because of the prognosis and the care that that child needs. So I think in that instance, which is relatively uncommon, it makes sense to test for the Huntington's gene in the child. The tricky part is deciding when a child is having possible symptoms of juvenile Huntington's, because children, particularly adolescents, can have a lot of acting out and abnormal behavior, and that doesn't necessarily mean they've got symptoms of Huntington's. So a key thing is, how are they keeping up physically with their peers, both physically and intellectually in school, and is there clearly a drop-off in their performance from better to worse that's consistent and ongoing? That's usually the best clue to juvenile Huntington's. Another common question is, does the age of onset of this disease run true in families? And families often tell us that it does, and they're right to a certain extent. And that's because of the characteristics of the inheritance of the CAG repeat expansion. The most common thing that happens when the expansion is transmitted to a child is that it stays the same size. So if you have a family where the repeat expansion is relatively small and it correlates with a relatively late age of onset, that's going to be fairly consistent in the family. On the other hand, if you have a family where the expansion is quite large to begin with, it tends to be large in everyone in the family, and the age of onset tends to be earlier. So in fact, it's fairly common for age of onset to be relatively the same within a family. However, there are always exceptions, because that CAG expansion can get larger when it's transmitted, or occasionally it can actually shrink and get smaller, and so the age can vary, sometimes substantially, within various members of a family. Another fairly common question is, does Huntington's disease skip a generation? And the answer is, it can certainly appear to skip a generation. And the way that happens is that someone has inherited the mutation for Huntington from their parent, and the parent had signs of the disease, but the individual who inherited it dies at a relatively early age of something else, like a heart attack or cancer or a motor vehicle accident. And so they actually carry the mutation but did not live to an age that they would express symptoms of the disease. And yet if they had children, those children would be at 50% risk. And so it can look like it skipped a generation, but in terms of the repeat expansion mutation, it really hasn't. It just appears to be that way. Another question is, what is pre-implantation diagnosis of HD? So it's actually a major piece of genetic OBGYN technology these days, and that is that it's a method for determining if an embryo carries the mutation for HD. and selecting embryos that do not have the mutation and placing those back into a woman who then can have a pregnancy with a baby that's known not to carry the gene. So it requires in vitro fertilization of the egg from the mother and sperm from the father, and one or the other has the mutation for Huntington's disease, and then at approximately the 8-cell stage of the embryos, removing one cell and determining whether that embryo has the Huntington's mutation or not, selecting an embryo that does not, putting that back into the mother, and hoping it will proceed to a pregnancy and the birth of a child who doesn't have the mutation. It's an amazing piece of technology, and I think it's going to become more and more value as time goes along and the technology becomes even better. But it does have some problems. One problem is it's quite expensive. It typically costs $15,000 to $20,000 to have the whole process of in vitro fertilization plus the pre-implantation diagnosis. Another problem is it doesn't always work. So approximately 70% of the time, 60 to 70% of the time, the embryo doesn't take and no pregnancy results, and that requires repeating the process again at expense. So it doesn't always work, and the technology probably will improve and become less expensive in the coming years. So if other persons out there have some questions that may have occurred to them, please feel free to sign in and let us know what that question may be. I should note that the pre-implantation diagnosis is different from prenatal diagnosis during doing amniocentesis. There, the fetus has already developed to the 15 to 20 week stage, and you're simply determining by amniocentesis whether or not the fetus carries the mutation for Huntington's disease. And in that technology, one can only decide whether or not to end the pregnancy. Another question is, can genetic testing be done anonymously? So the strict answer is yes, it can be done anonymously, but a better answer is that it should not be done anonymously, in my opinion. I think it engenders too many problems with the testing process. You don't know who you're dealing with. You don't really know the accuracy of the family history. We usually try to document that someone in the family has had a positive genetic test so we know we're dealing with the correct disease. And so anonymous testing is, in my opinion and my experience, is fraught with dangers, and I avoid it whenever I can. You know, it's really important to realize that there are other diseases that can mimic Huntington's. They can be inherited, they can involve the brain, they can cause progressive degeneration, their onset can be in middle age, they can frequently be confused with Huntington's. It includes some forms of hereditary dementia, some kinds of frontotemporal dementia, some kinds of Alzheimer's disease, some of the hereditary cerebellar ataxias. They have become hereditary Parkinson's disease. They've been confused with Huntington's disease, and if you're not certain of the diagnosis in the family and you just do the Huntington's test, you may be testing for the wrong disease. And doing it anonymously can get you into those kinds of problems. Another question is, what is the life expectancy of a 40-year-old that has HD? So the life expectancy of Huntington's, that is the duration of the disease, varies a lot. And the general numbers that are given are that the average duration of disease after someone has shown the first symptoms of the disease is 15 to 17 years. So that's the average time interval that someone will live after they first express symptoms. But that's just an average. And the disease can be much more progressive, particularly with early onset. I've seen people die in less than 5 years of the onset of symptoms. And the disease can also be much more prolonged. I've seen people who have lived more than 25 years after the onset of symptoms. So typically a 15-year duration, so a 40-year-old would die in their mid-50s, but with a wide range around that. And the progression of the disease is more rapid in the juvenile cases, and the typical juvenile case only survives about 8 years after the onset of symptoms. So it looks like we probably have no more questions, so I want to thank you for attending this module number 2, the genetic factors of HD. Next week in module number 3 of HD101, Dr. Jane Paulson will discuss the prodrome of HD beginning at 6 p.m. Eastern Time. Immediately following that presentation, Dr. Paulson will discuss the cognitive features of HD beginning at 7:15 p.m. Eastern Time. If you have not already registered for these modules, please do so by going to www.hdsa.org/CMEHD101. Thank you very much for your time and attention. To obtain your CME credit for this module, go to that same website, click on the post-assessment link. You must score 75% on the post-content questions to obtain CME credit, and you must complete the evaluation. Thank you very much for your attention and attendance.